hard copy of notes on cell cycle. Okay, so first things first, the cell cycle starts here. You're going to need 6 colors. All right, there's 6 colors I'm going to use. Definitely, if possible, make one of those a red. And yeah. All right, so this is where the cell cycle starts. The cell cycle starts with a very small cell. Okay, so that right there is a small cell with its little nucleus. Now that small cell, okay, it is going to grow and grow and grow and grow during what's called G1. This is the first phase of the cell cycle. Okay, the cell grows bigger and all organelles replicate. So now we have a medium-sized cell at the end of this cycle. I'll come back to that later. Okay, so that medium-sized cell will then go from G1 into what's called S phase. S stands for synthesis. Synthesis means to make. What's being made here is DNA, okay? We are making DNA copy. All right, so in the process of DNA replication, you start with 1 copy, and you end up with 2 copies. And that semi-conservative replication. Okay, this is happening here. So right here we have 1 copy of each chromosome. And in that process of in the process of S phase, each one is going to get replicated. So we'll have 2 copies of each chromosome. So we start off with 1 copy. At the end of S phase, we have 2 copies. All right. Now, we'll go in from there to G2. Notice it's G1, S, then G2. 1 is not immediately followed by 2. It's G1, S, G2. We start off with that medium cell. And we end up with a large cell. Okay? That large cell now needs to be divided. Okay? So during G2, the cell grows even bigger, preparing for mitosis, which is the cell division, by making enzymes. So it makes the enzyme necessary for which is the next step, which is the cell mitosis. All right, mitosis is the cell division. This whole crazy thing right here, this piece of the pie is all mitosis. Okay, so there are, the end of the cell cycle is what mitosis is. 1 parent cell will become 2 daughter cells. So you'll end up with 2 of those small cells at the end of this. The daughter cells are identical genetically in every way possible. All right, the stages in order. There are 4 inner stages of mitosis. So we have prophase, metaphase, which is written in for you because it's a small space, anaphase, and telophase. Okay, those 4 make up mitosis. After mitosis, we have cytokinesis, which is the final dividing of the cytoplasm. This is dividing the nucleus. Mitosis is dividing the nucleus, the DNA, whereas cytokinesis is just dividing up the organelles and dividing the cell literally into 2. Okay, cutting off the cytoplasm. All right, now let's do a little bit of coloring here so we can clarify what's going on. So we have here, if you have red, color the little stop signs red. These are the checkpoints. Checkpoints are places in the cell cycle, little stop signs there, where the whole thing just pauses for a moment to check to see if there are any errors, okay? So checkpoints. If there is an error, those errors get fixed. If the error cannot be fixed, the cell will do what's called apoptosis, which is cell suicide. The cell will kill itself because it can't fix the problem so that it doesn't become a cancerous, problematic cell, okay? So those checkpoints make sure that the cell is doing what it's supposed to do and growing and doing the processes it's supposed to do. All right, back to the beginning of the cell cycle. Gap 1, G1, all right? That is starting right here, beginning of the cell cycle. This is G1. Okay, it doesn't have to be the marker, you can do this color pen. You can do this with the color pencil, that's fine. Okay? Synthesis. The making of the DNA immediately follows G1. We start off with 1 copy of every chromosome and we end up with 2 copies of every chromosome. This is S phase. This is G1. Okay, DNA is replicated there in S phase. All right, G2. The cell grows even larger and prepares the enzymes it's going to need to do cell division. Okay, so that's G2. Grows nice and big, ready to be divided. Now, interestingly enough, these 3, gap 1, synthesis, and G2, these all together, okay, so S, G1, G2, all together, this is where the cells spend most of their life, and this is called interphase. Okay, nice. Almost blacked that out, oops. Okay, so all 3 of these together, that is interphase. So you see this arrow right here in the middle? This is all interphase. Okay, all 3 of those cycles together make up interphase. G1, S, and G2 combined. Okay? And then we have the very end, which is mitosis. So the prophase, metaphase, anaphase, telophase, that is mitosis. Okay, this combined makes up mitosis. But M phase in itself actually is mitosis plus the cytokinesis. All right, now I've talked about everything but this corner over here. G0, gap 0. Sometimes a cell is done doing the cycle. It's done. It doesn't need to do anymore. It's become so very, very specialized that it's done dividing. Okay? So the cell that has exited the cycle, this may be temporary, it may be permanent. Some cells will exit the cell cycle because they don't have enough nutrients. They need to pause, waiting for there to be more nutrients to be able to continue the cycle. Other times it's permanent when we have like a specialized cell. The cell that is at rest or in stasis, there's a fancy word for being at rest. This cell is not growing. The cell is possibly never going to divide again. Like I said, this happens with some very specialized cells. That gap 0, that exit happens during G1. So if a cell is going to go to the G0 phase, it will exit during the G1 phase. Okay, and there you go. Those are your notes. on the first page of cell cycle.